Hello, this is Sally's Book Talk. Tune in daily for great books, bringing you the best from around the world. Today's book we're exploring is The Biological Cultivation of a Foodie. Yes, fellow foodies, this is the exclusive science book written just for you. I believe after reading it, your perspective on eating will go beyond just taste, aroma, and appearance. You'll evolve from an ordinary foodie into one with biological insight, historical depth, and philosophical thinking. After all, the book's subtitle is The Scientific Saga of Fat, Sugar, and Metabolic Diseases. First, let's introduce the author, Wang Liming, a biologist born in 1983. He's currently a professor at Zhejiang University's Life Sciences Institute, specializing in neural and lipid energy metabolism. He earned his bachelor's from Peking University's School of Life Sciences, where he formed mentor-friend relationships with renowned scientists like Rao Yiwang, Wang Rodong, and Shi Yigong. He later pursued his Ph.D. at Caltech under Professor David Anderson, a leading figure in Nanso research. Having interacted with top-tier biology masters globally, he naturally gathers vivid anecdotes. Professor Wang Liming is perfectly suited to narrate the scientific saga of fat, sugar, and metabolic diseases. The author argues that after enduring long-term food scarcity, humanity has finally entered modern society. Our intense desire for food is an innate instinct. Thus, in our abundant modern world, everyone is essentially a foodie. So what's the biggest concern for foodies?
Exactly, obesity, along with related issues like high cholesterol and diabetes.
This book explores the stories behind these diseases, but it's not a health guide. The author also avoids giving specific advice. In this book, Professor Wang details scientists' research on metabolic diseases and the stories of fat, cholesterol, and insulin behind them. At around 200,000 words, we'll break it into 3 parts on biological knowledge every foodie should know. Part 1. The story of fat behind obesity. Part 2. The story of cholesterol behind high blood lipids. Part 3. The story of insulin behind diabetes. Following the book's order, we'll start with fat. In this section, you'll learn how biologists study fat, how our bodies regulate it, and what scientific weight loss methods exist. Perhaps their research findings will strike you as both mind-blowing and enlightening. Let's first discuss how scientists discovered fat regulation mechanisms. In fact, the scientific study of fat has a surprisingly short history, given that food scarcity has accompanied most of human history. For millions of years, truly obese individuals were rare, making fat an understudied subject. It wasn't until mid-20th century that Dr. Douglas Coleman became the pioneer in this field. How did he establish this research domain? Let's revisit 1958. 27-year-old Dr. Coleman had just earned his biochemistry Ph.D. from University of Wisconsin-Madison and joined Jackson Laboratory. Jackson Laboratory bred various strains of lab mice. Coleman noticed 2 voracious eaters among the mice, exceptionally large and obese specimens. Indeed, their abnormal size made them impossible to ignore. Scientific curiosity ignited. Why were these mice so ravenous? Coleman determined to investigate, naming them fat mouse and sugar mouse for differentiation. But how to study them? In that era, with no existing fat, research, or precedents to follow, Coleman didn't examine digestive systems or enforce exercise. He hypothesized, could these mice lack something in their blood that regulates appetite, causing overeating? The crucial question: how to prove this? You might suggest simply transfusing normal mouse blood. But Coleman took an unconventional approach. He surgically connected obese and normal mice through their dorsal skin flaps. As wounds healed, their capillary systems became interconnected. Far more effective than simple blood transfusion, wouldn't you say? After painstakingly pairing obese mice with normal ones, Coleman observed perplexing results. Contrary to expectations, the connected obese mice didn't reduce eating or lose weight as predicted. The experiment showed only fat mouse met expectations, quickly dieting successfully, while sugar mouse kept eating normally. Strangely, its conjoined partner lost appetite and starved to death. What happened? After days of contemplation, Coleman had another breakthrough idea. He believed his theory was sound, but needed refinement. The mice's appetite wasn't just about blood deficiencies, but also related to substances in bodily tissues. These two factors must interact, both needed for normal appetite regulation. Under this theory, fat mouse lacked blood factors, but had tissue factors, while sugar mouse had the opposite. If correct, conjoining them would make fat mouse slim, but leave sugar mouse unchanged. After Schulende skin suturing, Coleman observed the conjoined obese mice. Fat mouse slimmed again, while sugar mouse remained unchanged. With hypothesis confirmed, an excited Coleman began hunting the mystery substance.
Unfortunately, Coleman retired without identifying the substance.
In 1994, Dr. Jeffrey Friedman finally solved Coleman's puzzle. Interestingly, Friedman began this research fresh from his PhD, now in the molecular biology era. Friedman believed Coleman's biochemical approach was flawed, advocating molecular methods instead. Here's a brief explanation of these two methodologies. Biochemistry, developed earlier, views organisms as chemical mixtures. To find chemicals, tissues are homogenized into slurry, then purified for study. Molecular biology emerged after DNA structure and central dogma were established. It centers on genes controlling proteins. Finding a protein means finding its gene. DNA sequences, combinations of 4 bases, enable standardized analysis without complex purification. If you're still unclear about molecular biology, that's okay. With advanced methods, Friedman simplified Coleman's problem. Just find the missing genes in both mouse types. Though the strategy was simpler, Friedman still spent 8 years on this research. Yes? 8 full years! Mice have over 30,000 genes to examine. So what was this mysterious substance? It turned out to be a protein that slimmed obese mice, named leptin by Dr. Friedman. How is leptin produced? Surprisingly, it's generated by our white adipose tissue. Yes, the very fat in our bodies produces this weight loss hormone. Soon after discovering blood leptin, scientists identified leptin receptors in the body. This was the missing component in sugar mouse. Why was fat mouse obese? Fat mouse lacked the leptin gene, making it unable to produce this hormone. Supplementation led to weight loss. Why was sugar mouse obese? Sugar mouse lacked leptin receptors. It couldn't sense leptin levels, regardless of supplementation. Remember, sugar mouse is dead? Conjoined partner.
Excess leptin from sugar mouse suppressed its partner's appetite, causing starvation.
Fat serves as our body's energy reservoir. When we eat more, increased fat produces more leptin to suppress appetite. Reduced fat means less leptin, signaling the need to eat. This reveals the sophistication of our body's regulatory system. How does understanding fat regulation help with weight loss? This is probably your biggest question. After Friedman's 1994 discovery, scientists and pharma companies saw leptin as the ultimate diet drug. Obesity was becoming a major health concern at that time.
But after 16 years of effort, clinical trials declared leptin ineffective for weight loss.
Actually, most of us don't lack leptin genes or leptin sensing mechanisms. Excess leptin fails because of leptin resistance. The body becomes insensitive, especially in obese individuals. The causes of leptin resistance remain unclear. You might think after decades of research, is leptin useless? Not at all. For rare individuals born without leptin genes, it's life-changing. Cambridge University, 1997, and NIH, 2002, both confirmed leptin's therapeutic effects. But for most people, obesity involves complex factors beyond leptin alone. Scientists still have much work to do. Let's explore weight loss from another angle. Fat energy serves three purposes: metabolism 60%, physical activity 30%, and digestion 10%. This means exercise can't match metabolic consumption. So how to approach weight loss? Control intake, boost metabolism, plus exercise. Three-pronged approach. First, reduce intake via surgery, stomach expanding capsules or enzyme blockers. Second, enhance metabolism, make the body burn more energy. And scientists found brown fat activates in cold conditions to burn fat. Brown fat cells contain abundant mononutrient for fat burning. Normally dormant brown fat activates in cold environments to generate heat. So style over warmth might have scientific merit after all. We can strategically use cold exposure for weight management. This field is still emerging with many unknowns. Professor Wang believes true weight loss drugs will emerge from this research. In summary, fat produces leptin to regulate itself, but we can't use it directly for weight loss. Fat energy allocation, metabolism, activity, digestion provides weight loss insights. Now, let's discuss cholesterol. Cholesterol research predates fat studies, but progressed slower. First, documented in 18th century, French doctor extracted it from gallstones. Hence the name cholesterol. You might know its relative, hyperlipidemia.
Hyperlipidemia, excess blood fats, silently causes coronary cerebral diseases. These silent killers strike suddenly without warning symptoms.
From lipid imbalance to cardiovascular events, decades-long hidden progression. Direct correlation, blood cholesterol levels, and heart disease incidence. Cholesterol is essential for cell membrane structure. If excess cholesterol harms health, where's our regulatory mechanism? First research phase, cholesterol biosynthesis pathways. Dr. Konrad Bloch discovered hepatic cholesterol synthesis from acetate. This earned Bloch the 1964 Nobel Prize in Medicine. Goldstein and Brown discovered skin cell cholesterol production and HMG-CoA reductase. They proved LDL cholesterol's feedback inhibition mechanism. Puzzle remained: how does packaged cholesterol enter cells to signal stop? Research continued. Enter Dr. Nils Thunstrom, treating a young patient.
The boy had 6x normal cholesterol, LDL levels with severe angina.
Dr. Stone was also helpless. Fortunately, Dr. Stone followed biological research and sent the boy's skin samples to Goldstein and Brown. The duo discovered the boy's cells overproduced cholesterol with serum present. This indicated normal cells, but defective serum regulation. They hypothesized LDL couldn't enter cells, discovering cellular ingestion mechanisms. Students isolated LDL receptors, confirming genetic receptor deficiency. Akira Endo discovered mavastatin from fungi inhibiting HMG-CoA reductase. Remember this crucial cholesterol synthesis enzyme. Mavastatin revolutionized treatment. Pratil developed Lipitor based on it. 2003, Franco-American team validated theory via PCSK9 gen defects. 2015 saw PCSK9 Findine's launch. Scientific legacy cemented. Insulin. Story begins. 1889 French found pancreas removal caused diabetes. 1901 Eugene Opie linked pancreatic islet damage to diabetes. I halted European research breakthrough came from Frederick Banting. Banting differed. Average student, barely earned bachelor's degree. Canadian war medic turned Toronto University lecturer. At 29, Banting read about pancreatic duct ligation and had an epiphany. Professor MacLeod provided resources despite Banting's lack of credentials. Experiment. Pancreatectomized dogs. vs. islet. Extract injections.
After 91 dog sacrifices, diabetic dogs showed improvement.
MacLeod and biochemist Collip successfully isolated insulin within a year. In 1922, insulin was first used to treat a critically ill diabetic child, restoring blood sugar to normal within a day. A medical miracle. Toronto General Hospital overflowed with patients. The 1923 Nobel Prize was awarded to Banting and MacLeod by the Swedish Royal Academy. Winners shared the prize money 4 ways and licensed insulin patents for $1 to enable mass production globally. Later discovery. Oral glucose stimulates GLP-1 protein secretion in intestines, triggering insulin release.
Type 1 diabetes requires insulin supplementation. Type 2 remains unsolved due to insulin resistance. Scientists' road ahead is long too.
The insulin story. Pancreatic islet research revealed insulin's blood sugar regulation capabilities for diabetes treatment. Our gut's sugar detection system regulates insulin release. While diabetes medications exist based on current understanding, research still has miles to go. The biological cultivation of a foodie chronicles metabolic disease breakthroughs through fat, cholesterol, and insulin narratives.
How we utilize these scientific discoveries to understand diseases, develop treatments, and protect health. Thanks to scientists' tireless work, we receive accurate diagnoses and treatments when ill. This book reveals the elegance and power of scientific inquiry.
That concludes our exploration of the biological cultivation of a foodie. Find keynotes below. See you tomorrow.